Hello and welcome to part 3 of this 3-part lecture series on microbial growth. In this part we will finish out this chapter, and we'll be looking at different types of media and then ways in which we can count bacteria and how many there are in a sample, for example. So let's get started here with selective media. Selective, and there is another type of media called differential media. Those two often go hand in hand, selective and differential. But I'll address them each individually so that you can better understand what the term selective means and what the term differential means. So selective media are used to detect the presence of specific microbes, usually pathogens. And so how the selective media work is they contain nutrients in them, compounds in them, that will suppress unwanted microbes and encourage the growth of desired microbes. So for example, the bottom picture there is showing this example. Bismuth sulfide agar is used to isolate the typhoid bacterium. There are others as well. Ones that we commonly use in my micro lab would be mannitol salt agar, which has a high salt content, and because of that salt content, it's perfect for growing Staph aureus. There's another one called EMB, eosin methylene blue, which contains dyes that inhibit gram-positive bacteria and encourages the growth of specific gram-negative bacteria, especially E. coli. So there are others. They are selecting, and that's why they're called selective, because they're selecting for the growth of a particular species. So they have stuff in them that's going to encourage the growth of desired microbes, the ones that you want to grow, and then at the same time then it's suppressing the ones that you don't want. And you can use it to detect for the presence of these pathogens as well. So you take a sample of somebody's their urine or their sputum or whatever, and you put them on the selective media, and if you suspect them of say having a strep infection, for example, you can put them on. You can put their sample onto a selective medium, and that encourages the growth of strep. And if strep appears, then there you go. It's been confirmed. If not, then something else. So we can use these then to kind of pinpoint and narrow down what a patient happens to be infected with. Differential media, often differential, it yields a color change. And so as a result of that, you can differentiate between colonies. So it makes it easy to distinguish colonies of the desired organism. So I mentioned earlier eosin methylene blue, or EMB. It contains lactose, which is perfect for E. coli because E. coli ferments lactose, and it's selective in that way, and it's differential in that the E. coli, when it grows on the EMB agar, which is naturally a maroon color, when the E. coli grow on it, the bacteria thrive. They produce a compound that actually causes them to turn this metallic green color. No other bacterium will do that, will turn that green color on EMB. So that color change enables you to differentiate E. coli from other bacteria that may be growing on the EMB, because other bacteria can grow on EMB, but they don't produce that metallic green color on their surface. So selective and differential almost always go together. The media have compounds in it that select for particular species, and then they also can enable you to differentiate between species, usually because of a color change that occurs. Either in the agar, or in the case of like EMB, the color change occurs in the bacteria. Then there's enrichment media. Enrichment media encourages the growth of a desired microbe that is far outnumbered by other species. I'll read the example because it explains it very well. The example, once you read that, then it makes sense. Culturing, for example, nitrogen-fixing bacteria. There are very few nitrogen-fixing bacteria, so we can use enrichment media to culture them. Bacteria capable of transforming atmospheric nitrogen can be isolated by incubating a soil sample in a culture medium which has all of the ingredients necessary for growth except nitrogen. Only those bacteria that can fix nitrogen will grow, and those that can't won't. So there, like I mentioned earlier, there are very few bacteria that can carry out the process of nitrogen fixation. And so the bacteria that can't, they will not grow on this enrichment media because there's no nitrogen in it. And so they will die off. They have to have the nitrogen in order to live. And then the nitrogen fixing bacteria will thrive because they don't rely on the nitrogen in the agar, or I should say agar, they don't rely on the nitrogen in the medium. They get their nitrogen from the air. And so they'll grow and they'll thrive. Pure culture, and as the name indicates, it's only one species or strain. It's a pure culture. There's nothing else that's tainting it. It's just one single species. And so this is what you want to achieve in a micro lab. You want to observe, experiment with usually just one particular species. And so your goal is to obtain pure cultures. And then you don't have anything tainting it. You don't have anything that is competing with the bacterium that you want to experiment with and research. So you want to achieve pure cultures and obtain pure cultures. A colony population of cells, I mentioned that earlier, a group of thousands of cells growing on a particular medium. And a colony is often called a CFU, or a colony forming unit. One way to get a pure culture is by using what's called the streak plate method. And there are a few different streak plate methods. My favorite is the quadrant streak. And what happens is you start on one area of the Petri plate, you streak the bacteria on one section, then you sterilize your loop, which is the instrument that you use to transfer the bacteria. You sterilize it, and then you do, if you look on the picture on the right, if you look at that Petri plate on the lower right area, that would be quadrant number 2. So quadrant number 1 would be above that, and then you would sterilize, then you would drag the bacteria out of quadrant number 1 into quadrant number 2, sterilize your loop again to kill any bacteria that might be on it, then drag bacteria out of quadrant number 2 to make quadrant number 3, and then do it again for quadrant number 4. And then what you end up with are little tiny dots of bacteria, little colonies. Those are pure colonies. And then you can transfer them to a new Petri plate, and then you only have one species. You don't have multiple species that are growing on the same plate. So the quadrant streak, or any streak plate method, but this is my favorite, is used to get pure cultures. How do they reproduce? By far, the main form of reproduction in prokaryotes is binary fission, where they just split in half. This slide here, I wanted to show you the other three that are not nearly as common: budding, kinetiospores, and fragmentation of filaments. So the upper right picture, that's just showing the budding process. The nucleoid replicates, in other words, the nucleic acids will double. The new nucleoid moves into the bud. Then you have what's called a young bud. I love that. That's such a cute little young bud. And then you have the daughter cell. It splits then. It separates from it. And so this is a form of reproduction called budding. Kinetiospores are on the lower left. They're called kinetiospores. Spores are reproductive cells. Kinetia are the stalks on which the spores are attached to. So the stalks kind of move them away and then enable them to spread out in an environment and then colonize new areas. Fragmentation of filaments, they just keep splitting. It's kind of like mold-like growth. And so sometimes whenever you're looking at a culture, sometimes you can be mistaken and think that that is a mold because it has mold-like growth, but this is another way the bacteria reproduce. They just keep sending out new filaments. They have filamentous growth. And then binary fission, since it's the number one way, it gets a whole slide to itself. It's so exciting. It's not all that exciting. The DNA, the nuclear, the nucleoid, it basically separates. It's very much like budding, except instead of sending a young bud out the side, the one end of the bacterial cell, it just splits right down the middle. And then you end up with two cells. So they have the identical DNA. All forms of reproduction in prokaryotes are asexual. There is no combining of DNA from two cells. There's no sexual reproduction in prokaryotes. So all the four that we looked at here, these four forms of reproduction, are all asexual. They're just making clones of themselves. A typical bacterial growth curve. So if you were to look at the number of bacteria as they grow in colonies, this is the growth curve they would kind of follow. You have a lag phase at the beginning in the lower left. Lag phase just means the numbers aren't increasing. They're just they're alive and they're maturing, but they're not increasing in number. And then, if all the conditions are optimal, you'll go into the exponential growth phase, where one bacterium becomes two bacteria, two become four, four become eight, eight becomes 16, and they can double their numbers if conditions are optimal. Some species can double their numbers about every 20 minutes. So you end up with an enormous number of bacteria in a very short period of time. Eventually, the environment cannot sustain exponential growth. I mean, exponential growth cannot go on forever. So eventually it'll level off. It's what's called the stationary phase. And then it'll reach the death or logarithmic decline phase. And that would occur whenever all nutrients that are available in the environment have been depleted. There's nothing left for them. And so their numbers will exponentially decline. So they exponentially can increase, and then they exponentially decrease at the other end as well. During the log phase, bacteria are most susceptible, the exponential growth phase, that is, because they are so metabolically active. There are so many of them that are, and so that becomes the primary function of the bacteria during the log phase. So they're most susceptible to them. Susceptible to what? To disinfectants and antibiotics and antimicrobials. During the death phase, a process called involution occurs, which what happens is dramatic changes in their morphologies or their shape. That's what morphology is. There are dramatic changes in their shape. Well, what does that mean? It makes them difficult to identify then. And so identifying bacteria can be very important, knowing which microbe or pathogen you're dealing with. So during that decline phase, identification becomes difficult because of the process of involution, the process of them changing their shape. Counting bacteria. This is one way of counting bacteria. It's called the serial dilution. It's the most frequently used method for counting bacteria. So you take, if you look in the picture and if you follow it from left to right, you start with your original inoculum. How would this be used, practically speaking? Well, in the lab, I mean, I give my students like putrid milk, for example, sour milk that's just thriving with bacteria. We take a milliliter of that rancid milk out of the container and we put it into 9 milliliters of this picture shows broth. We use distilled water, sterile distilled water. And then we have 10 milliliters total. So our dilution is a 1 to 10. 1 milliliter of nasty milk loaded with bacteria to 10 milliliters total in that test tube. Then out of that test tube, we pipette 1 milliliter of that solution and squirt it onto a Petri plate. Then we take a 2nd milliliter out of that first test tube and we pipette it out from the first test tube and put it into the second test tube. Now we have a dilution of 1 to 100. Then we pipette 1 milliliter out of that tube and put it onto a Petri plate, and then a 2nd milliliter goes into the third test tube, and so on. And what happens is you're diluting the number of bacteria every single time in a series, and that's why it's called serial dilution. The purpose of this is to get the bacterial colonies to a countable, manageable amount, so that by the time you get to the end, it's not just solid growth of bacteria. I mean, you can't count anything. So you want them to separate out into their individual colonies. Then you can count individual colonies and get an idea about how many bacteria there are in a sample. In an ecological way, you could use this, say, for example, testing water quality. Taking water from a reservoir or from a stream that feeds a reservoir to then be used in the public water supply and do a serial dilution to determine how many bacteria are present in a particular water source. Another way to count is, and this would be direct measurement, is filtration. Used when quantity of bacteria is very small, like a pristine lake or something, or a stream. You have to have at least 100 milliliters of water. You pass it through a filter, which you can see in that micrograph on the left from an electron microscope. Those little holes in there, the little black circles, allow water molecules to pass through, but the bacteria can't. And this is most often used to detect coliform bacteria to see if a water supply is contaminated with a pathogen. And this is directly counting. So then you plate them and then you can count the cells and figure out how many there are in a particular sample. This is used by the local water authority. I had a microbiologist come and speak to my students once a couple of years ago, and she said this is what they use. They use the serial dilution, the previous slide, and they also use filtration as well whenever they take water samples from any of the streams that are feeding their reservoirs and from the reservoirs themselves. And they have to do this to meet government guidelines. So this has to be done on a routine basis. So they go back up into the mountains, they collect their water sample, they bring it back to their lab, and then they carry out these counting methods and then submit their data to the government agencies to prove that their water is drinkable. Another way you can do this is by putting them on a special slide and then looking at them under a microscope. And the slide has squares, it has a grid on it in which you can count bacteria. It's very tedious. You have to be very detailed and try not to lose count, otherwise you have to start over again. But this is a way to count directly counting how many bacteria there are in a sample. Again, just another way. It's like filtration, directly counting. This is just a different method of directly counting bacteria in a sample. To estimate the number of bacteria, this is an indirect method. You use a spectrophotometer. And so what the top part of the picture shows is you have to put a blank in. So you have a test tube with sterile distilled water. You put that in, and then that sets the standard. And then you take your sample in a test tube and determine how much light can penetrate it. Scattered light that does not reach the detector gets bounced around. Some light gets transmitted. So the lower the percentage, the more turbid your sample is, meaning there's more stuff in it. Now, would it have to be bacteria? Not necessarily. It could be other things that are within your sample, but it can be an indication of bacterial populations, and that's why this is estimating. This is an estimation. So more light shines through, less turbid. Less light shines through, more turbid. And this can give you an indication of the amount of bacteria that would be present in your sample. Two other ways, metabolic activity. So you can collect the amount of carbon dioxide. More carbon dioxide that is collected, that means lots of bacteria because the bacteria are metabolically active, producing more carbon dioxide. And so this is, again, an estimation. If there's a lot of CO2, that means there's a lot of bacteria. This would be something where you don't have to have an exact number or a direct count. Dry weight is another estimation method as well, used to measure the weight of filamentous bacteria or molds. Remember how I said filamentous bacteria, they grow like a mold? That's why we can use this dry weight. So you get rid of, you remove the specimen from your growth medium, you get rid of all the extra stuff that may be present in the material that it's growing in, you dry it out in a desiccator, which removes all the water and moisture, and then you weigh it. And obviously if it weighs more, that means there were more bacteria in your sample. Again, it's an estimation. It's not a direct count. Okay, that wraps up Chapter 6. That was this is the end of Part 3 of this 3-part lecture series. I hope this was of help to you and was of value to you as well in your pursuit of understanding microbiology a little bit better. So stay tuned for the next chapter. We'll be continuing with microbial growth and how to control microbial growth and ways that we can control their growth so they don't grow exponentially. And as always, thank you for watching.